Okay, good morning, students. Welcome back to the class, Lecture 10. Today we will be talking more about promoter prediction methods. As per the last class, we have seen a lot of stuff about gene prediction method, genome sequence analysis. If given a blank sequence of ATGCs, how do you predict what are the genes if given sequence is coding for a gene or not? We will briefly recap that information so that we can connect to the promoters here from the genes. Then we will go into what are the promoters and their features. And then we specifically talk about transcription factors and then promoter prediction methods, principle, and then databases. These two are again combined into the slides. So you have to make notes on your own in that. So coming on to last class, this is our golden slide for most of the genomics classes if you are looking for sequence analysis. We spoke that you look into blank sequence, given sequence, you deduct whatever you can see based on your understanding of fundamentals of molecular biology and genomics. Let's take whatever we have seen last class. We went into this slide with gene prediction methods. First, we spoken about open reading frames, then intron-exon bridges, polyadenylation sites. Based on these sequences within a given unknown sequence or a known genome sequence, you can deduce whether the sequence codes for the gene or not. And I might have missed these slides. It will be very useful for these flowchart diagrams. You can look into that. Given genome sequence, you take it, translate them into ORS based on the databases. This is called pattern prediction method that we looked at in last class. Or you can go with softwares available with increased efficiency, but accuracy may not be good with the softwares. You go with homology-based database because you might have a sequence already a similar comparative genomics you can do, and you compare with the sequences based on the homology. If the database available, you go with homology-based data gene prediction, analyze and detect the genes out of it. If you don't have homology specifically as such, not proper annotated genomes, then you go with ab initio gene prediction methods. You can locate the genes. And you can put input sequence, scan with a 3 codon sequence periodicity, and then another method called Fourier spectrum analysis that gives a sharp peaks in a given sequence, relative height of the sequence, uniformity of the sequences. You can code probable genes in that. So far, so good with the flow of the slides and recording. Is everything okay?
Yes, sir.
Yes.
Okay, perfect. So the Fourier transform is just nothing but you are looking at the frequency of nucleotides given in the x-axis. In the y-axis, there is a term called power spectrum. Based on that, based on the frequency of the nucleotide in the given nucleotide sequence, you will get a peak. The peak will tell you that there may be a consensus sequence, there may be a pattern of sequence, what you have put in the input. But based on the variability of the nucleotide sequence and variability of the variance in nucleotide distributions, for example, the Fourier spectrum may look like this, that because DNA polymorphisms, we have variable, you're looking for a human genome population, there may be polymorphic sequences that may give rise to different variability of the spectrum here. We have used again specific softwares that can help for looking into eukaryotic, prokaryotic genomes in terms of genome prediction, bacteria, eukaryotes, archaea, and viruses. So given a blank sequence again, so you now think of another question. So alternative approach, is there any sequence other than the gene segments, exon, introns, codons, or splicing? Other than that, is there more in this sequence? Yes. DNA sequences in a given genome, yes, there is always more elements into that, and more complex regulation allows for this complexity of the sequences and elements in that. What are those? We slowly will pick up basic understanding of promoters. We will go up with the what are the more sequences we are going to look in today's class. What are promoters in a gene, given gene? What are promoters? Gene promoters called. So this slide, you can look here. I will just first recap with the text and go and explain the diagram. Gene promoter, the DNA sequences located upstream of gene coding regions. So let's say start and stop codon here. ORF, open reading frame, first amino acid. And we already know from fundamentals of molecular biology that if this is a start codon, ATG starting from here, But transcription starts from upstream specific sequences called transcription start site. They don't start from here. They will go a little bit more in the upstream of the given sequence in the DNA. These are called DNA sequences, promoter sequences. And again, little bit more further, there is specific sequence for RNA polymerase binding. It all constitutes the part of the promoter starting upstream of this. They contain multiple sites. Those sites are responsible for binding of transcription factors in that to start a gene expression on or off. Some of the elements that present little bit close vicinity to the transcription start site are upstream of this codon. They are called as core promoter elements. And one of the element that is called consistently Tata box in eukaryotes. And further, you imagine that this is the basic understanding of the promoter here. You're looking at a kind of transcription translation happening a transcription of two strands only given a small stretch of DNA. Already transcription started, the promoter is here, the RNA polymerase is moving into that. And if you imagine as a whole genome, that the two strands in a much complex way, they are condensed into histones, and only histones will open only whichever gene they want to on. They will open specific fragments of histone and DNA complexes, other fragments will be off. But we will take this basic understanding again, little bit more broader, talk into individual concepts. This is, you are looking at a promoter or gene elements of generalized organization of the gene and regulatory elements. The upper slide is eukaryotes, the bottom is prokaryotes. You can see in the eukaryotic genome, again, you are starting from a start of transcription here. The exon may be here, for example, RNA start here, but the protein start ATG may be somewhere down. But we usually start the transcription with the denotation of +1 nucleotide that says the start of the transcription region. It goes with looking into the DNA sequence, 5' translated and 3' untranslated, 5' untranslated, exons, introns, RNA goes into that. This is part of the gene prediction we looked. But if you go into a little bit more, whatever I was told in the previous slides, there will be a core promoter region. Upstream of the core promoter, again, some proximal control elements. We will see again more depth into that. Location is, you are going in from the +1, you designate, you go into the upstream, you are talking about plus nucleotides. You go into the back, you designate as minus nucleated at downstream of the transcription start site. But the same designation in eukaryotes almost consistent, but there is variability we will see. In prokaryotic, typical organization is like this, -10 region, -35. So you go into individual, we are looking again eukaryotic gene, thymidine kinase gene, start site here, a Tata box at -25 nucleotide, a specific GC box, CAT box, again GC box here. If you look -30 base pairs upstream, there is an octamer transcription element that will have specific function. So why I am talking about these sequences is that, so these sequences will ask us to think one question. We will come back here again on this, what are those? You look into this promoter, look into the sequences, we can ask one specific question. All the promoters should look the same. Let's say eukaryotes, prokaryotes, and genes. And there is a universal mechanism, universal consensus sequence for the promoter so that everything goes on the same way. Yes, there is an on switch, only one sequence in all both eukaryotes and prokaryotes. That's it, you can switch on and off. Or there may have special ones, promoter have some special sequences. Or they, if then, what are the special about? What is the specialty about each sequence? Yes, promoters are not unique, they are not same in all species. Different to cells, actually they are different from each cell to cell. We will see why they are that. Now you zoom in with the sequences. What is the speciality of the each sequences? Because the each sequence corresponds to specific kind of proteins called transcription factors specifically. This sequence is consistent for transcription factor 2D binding. This sequence is specific for some other transcription factor. They have a specialty for unique binding of a specific protein that is called transcription factor. For example, Tata box located approximately from the start site, and again it may not be again a universal rule. It may be different. Some of the promoters don't have Tata boxes. We will see what are these, and they have a protein called Tata box binding protein. It's part of the transcription factor 2D complex, 2D part that will bind specifically here. The GC box binds to specific specificity factor, that's called SP1, and the CAT box, it binds to a specific CAT box transcription factor, CTF. And octamer, it binds to octamer transcription factor. So the sequences have a speciality, and only specific proteins will come and attach to the sequences in a specific position, specific orientation in terms of the nucleotides downstream, and then they regulate transcription. So that's why the promoter elements of a given gene or a gene sequence have been divided into two modules. One is called, the module is conserved in several promoters, that is called as, we will see what is it called. The module is shown to be binding for a specific transcription factor. So the conserved in specific promoter, that is called cis-acting elements in the DNA, given DNA, and then the transcription factor that bind to specific sequences and regulate the gene expression are called transacting factors. These are important terms. Any given promoter, we will talk about what are the cis elements in the promoter, what are the cis-acting elements, what are the transacting factors. Factors means these are the proteins involved in attaching to the given sequences. These cis elements are, what are the cis elements in more abbreviated definition form? Cis-acting molecules or elements act upon and modulate the expression of physically adjacent, operably linked polynucleotide encoding sequence. So what they are saying is that a given promoter sequence, given a DNA, given a start site, cis-acting molecules or sequences are a very close vicinity of the promoter region or start of the gene or gene coding regions, exons and introns. They are very close approximately. The transacting factors, they are mainly proteins. They are encoded by specific genes that may not be close to the gene of interest, where the promoter of interest you are looking. They are actually far away. They may be in other chromosomes also, because, for example, the transcription factor TF2D may be encoded by other promoter regions present in other chromosome, but your gene of interest may be in another chromosome. So cis-acting or close vicinity of the DNA promoter molecule. Transacting, they are produced from some other gene location, some other chromosomes, but they are really important for regulating the gene function. It's in general terms, this is you need to understand in general way rather than going into technical terms. For example, let's say this is the core promoter here, where the +1 start here is called as core promoter. We will see why it is called core promoter. Let's take an analogous example. The core promoter is myself, and my job is to, on the transcription or the gene's expression, that means my duty for the present scenario is to deliver a lecture to you. Then I am the core promoter. So my further dead end function is to give the lecture to you to make sure that you understand the class of genomics and proteomics. Can I do that on my own, being the promoter, and I can completely start on my own and then do a lecture for me only? And then that may not be fruitful. My gene expression is not to the 100%. I need you students in close vicinity. If given a class, we are doing the lecture. I am the core promoter standing on the dais. Your people are these cis-acting elements in the close vicinity of me, staying in the classroom, and you need me. I need you both. We start the lecture so that I can properly get the inputs from you, do the deliver proper lecture. So these cis-acting elements are yourself in a given classroom. I am the core promoter. And one more thing, for example, of the transacting factors in a given scenario now, we are going for online classes. Again, I am the core promoter. You are the cis-acting elements. We need to deliver a class properly, working in a coordinated fashion. But in an online class situation, without Zoom, without internet, I cannot deliver the lecture. See, that is the factor that is important for us right now. Though the promoter is here, myself, though you are here, unless you don't have a proper internet connection, unless you don't have a Zoom availability, we cannot deliver the lecture. So the Zoom, those are the factors, internet and Zoom, they are the factors of transacting factors. So a combination of Zoom, combination of yourself, vicinity of the core promoter, myself, we deliver a lecture 100% in a proper fashion. Is nothing else but nothing different from in terms of the talking gene expression in DNA, cis elements, close vicinity of the core promoter, and then transacting factors. There are other because I cannot control, right? Core promoter cannot control the transacting factors. It has to be controlled by Zoom company, controlled by whatever internet company I am looking for my network. So transacting factors, they may not be located in the close vicinity, but they are very much important as the sys elements. That is the system transacting element. So transcription factors, in terms of what they actually do, they recognize specific target sequences in the DNA and interact with other transcription factors also. For example, given the Zoom company, given the network, they have to work in concert. So there are many factors involved in that. Specifically, the transacting factors, how they control gene expression, they control in several ways. The factor may be expressed in a specific tissue, spatial regulation, specific tissue manner, specific time in development, temporal regulation. It may require specific modification, that is phosphorylation in per se. They may be activated by a ligand, or they may act constitutively. Constitutive means they don't need ligand. They can be on themselves, the receptor, and then they may be sequestered into a specific position, and specific environmental signal makes them to come out and attack to the nuclear DNA and start the gene. So transcription factors. And for example, in the promoters and transcription factors work in concert, and they are the one of the important technological inputs that we use for many molecular biotechnology applications. The promoters can be of a constitutive, without the ligand, they can be on tissue specific, tissue eye, tissue hand, inducible promoters given environmental signal or given an external signal that can induce a specific promoter when the external input is there. Then synthetic promoter, you use that. We may use it in the technology method and methods and explanation of genomic and proteomic methods. We may use this and expand it in a better way. But for the present class, try to understand the fundamentals. Again, going into a little bit into gene regulation, there are switch on and off molecules. They are called activators. They are repressors. When activator is present, the signal is on, the gene expression is on. Repressor is present, the signal is off, there is no gene expression. How do you look into molecular level? Prokaryotes and eukaryotes, there is an activator protein. There are specific elements in the promoter. They bind to that, gene is off, on. Gene expression, repressor protein, gene expression is off. That on and off varies from structural in terms of coordination. It varies from prokaryotes to eukaryotes. Eukaryote histones are there. Different enhancer elements will come. We will see about this. What are these enhancer elements? This probably not present in a prokaryote as such. So there is an enhancer that efficiently allows this complex to regulate in a coordinated manner. So if you look in a specific given gene, now we are not looking at the exons and introns. We are looking into the promoter elements upstream of the start site. There may be enhancer region here, promoter region, other boxes are here. There may be a silencer region. These are upstream activator elements that nothing but core promoter and proximal elements. We will zoom in them a little bit in the next few slides. Remember that enhancers may not be in the close vicinity. They may be further far away. They may be sometimes 50 kilobases far away, sometimes 10 kilobases far away. And they are also important. That's why they are called in terms of one more abbreviation we will see. But why I'm talking about these elements, these boxes, because when you want to predict a specific promoter of a given sequence or given genome, you can either use comparative homology study, you can blast off a specific family of the gene that you expect from unknown to specific family that looks similarity here, consensus sequences, putative genes here. Transcription start site, you do a blast sequence analysis, you can get elements that are GC box here, the cat box, there may be a consensus here. But again, the consensus again is a variable again. You may see that upstream elements and their position from start site may differ. For example, Tata box, there is a slight variability here in the position. And if you see cat box variable, there is no cat box here at all. And GC box, some of them they don't miss. So you look into this consensus, you can predict something out of it. And one more important features in terms, for example, Tata box less promoters. In certain given case, there is no Tata box in the promoter, but there may be GC and cat box are conserved. But how it happens, the transcription in that, the promoter will have a specific sequences called initiator sequences, INR. they are present in a kind of consensus here. The A will be always preceded by the C in the given sequence. There will be a pyrimidines, 3 pyrimidines, 5 pyrimidines consensus will come. But remember that it's not a Tata box, it's an insertion sequence, INR sequences that also helps in initiating the transcription. Yes, now you look into the blank sequence in terms of going into promoter and a combination of gene coding open reading frames, and a combination of both. You can look and predict whatever you want on your own interest. So in summary, the promoter elements have been divided into, this is the ORF, the gene prediction, going into promoter prediction, core promoter, proximal promoter, distal promoter. Core promoter, mainly cis-acting sequences embedded in the core promoter. Proximal elements, like, excuse me 1 second. Okay. Okay, hopefully you have read the abbreviation here about core promoter elements, proximal elements, distal elements. The distal may be, for example, enhancers may be in the distal, and the proximal elements are here. That maybe we'll see more detail. What are those? Core promoter elements can be divided into INR sequences, DRE, DPE, Tata box, DCE, and MTE. We will see what are those. And proximal elements can be divided into GC box, CAD box, and octamer. And you look into that when you're running a software, you're looking into that location of the genome. What are these? What are the proteins, consensus sequences in prevalence, in terms of prevalence in humans and other species. Remember that Tata boxes in humans here, they are only 12% in a given genome. So there may be other elements that are important for specific other genes. So in terms of abbreviation, what are BRE? B recognition element, downstream core element, downstream promoter element. This is a motif 10 element that is downstream of the promoter that promotes transcription of RNA polymerase. Remember that one more thing we are looking of downstream of the core element, upstream of the core element. That you see in this diagram, you are seeing some of them minus, right? Minus regions BRE, but this DPE and the DCE are up plus. They are not located in terms of the transcription start site here. These control elements for this core promoter element, they may be vicinity of the upstream from the +1, -40, these elements, or they may be in the downstream. Still the ATG is not here located. The ATG may be further away. So it may be located downstream, located upstream. That's why +40 and -40. Look into further more details about the elements here. Given a Tata box, 10 to 15% of human genes only. And the both DP and MT are downstream. They are in the plus region. That is also an important for promoter prediction method. In summary, metageovan regulatory modules controlling transcription. There are different cis-acting modules present here, proximal distance. There are many transacting factors, transcription factors that bind to specific region, and coordinately they will regulate the gene expression. That's why promoter regions are very important. If by mere presence of the in a sequence, the exons and introns and other sequences for protein coding regions, you cannot code any gene, you cannot switch on any gene, or you cannot give a specific protein in that. You need it specifically, you need a different concepts of promoter elements or regulatory elements. If you zoom into the enhancer that may be 50 kilobases away from the given promoter, they can loop into specific formation and they can combine with a specific complex of transcription factors activator. Enhancer always associates with the activator that makes the gene on. And if you zoom in terms of the histone region, how the distal and proximal promoters are there, you look into that. A core promoter here, ORF, and start site here, RNA polymerase binds, and then specific proximal elements here, and then the distal promoter elements far away from in the histone regions. Then remember that whenever there is always few genes are on, few genes are off in a given scenario. So the histone unwinds and it opens up the DNA and mainly promoter elements to come and associate with a specific protein and on the gene transcription, gene expression. Okay, thus coming on to the conclusion, how do you predict a promoter in a given sequences? It is based on the understanding of consensus sequences and specific in the location of the core promoter, other elements, distal promoters, and proximal elements, and also transacting factors given in transcription factors. You can deduce or predict a promoter regions of unknown genes or known genomes given a species comparison. How do you do that? There are, again, we will go into a kind of databases as discussed in the genome sequencing databases. Again, to a specific functionality, specific question of interest, there are different databases available. This is a specific database called eukaryotic promoter database. This concerns mainly different species of the eukaryotes. How do you want? What do you want to look in a specific given promoter region? You can use a different species, follow the guidelines, and then you can find the promoter regions and the sequences associated with it. This is for nematodes, C. elegans promoter database. Again, you give a given sequence of the whatever patch of gene of patch of genome DNA you are interested in, you can put it. Again, you see that you are starting from -500 or you are going into +100 of the given sequence, starting from the codon site +1. For enhancers, you have to go to 50 kilobases. That's why there is specific software that can be used for enhancer region only. This is in general of proximal regions and then downstream of upstream of the core promoter. Again, another DBTBS. This is another database that helps to find specific transcription factors of a given family, because transcription factors will have helix turn, they have specific motifs, and you can use a predict for hexameric motifs, predicted terminators, operons, regulons. This is another software that you can use for that. And one more software is called Regulon database. Regulon can use for regulators, and this is specifically used for prokaryotes, especially Escherichia coli and few other prokaryotic species. And you can find what are the promoters of the given prokaryotic species at the time. One more important database for model organisms, that is called Drosophila, Berkeley Drosophila Genome Project. This is also representative of data sets for human DNA sequences. You can compare with the humans given the promoter and splice site. You can also look into EST sequencing, Drosophila genome here. You can look into promoter and prediction of specific species and compare it with the human species for using this database. One more database specifically called PlantPan 2 version. It's used for specifically for promoter analysis of plant species and transcriptional regulatory networks of plants. You can read into that, how you can, there are many transcription factors, binding sites, you can find with that. So given a database, given an algorithm, given a software, how do you detect how the system works? Basically, it works through this flow chart. You have to design the algorithms before you put the databases or what you want to do. Your question you are looking for specifically is that you are trying to find the promoters of a given genome sequence or a given DNA sequence. So first, what you will do, you have to create a benchmark data set. How do you create the data set? You go, if given already you are working with a known organisms, you go with the promoter database, put the data if a specific sequences you are looking for into that, specifically relevant to the promoters here, nothing else. And then you might have to give a control to the system that if it is not a promoter, how can I identify? Then you can give a region of the non-promoter, for example, introns, exons, splice sites, polyadylation sites. You can give that control to the system also. A benchmark data set will be given. You refine the data. You give a specific features, specific motifs here. Select the feature and predict it with the model. What is the streamline in terms of non-promoter, promoter sequences, and match it with the given sequence you are testing on. Assess it, cross-validation, assess with other factors, and implement the tool so that a specific promoter prediction software can be deduced. What are the models specifically used in the promoters? One important stuff in the promoters is that a lot of the sequences are consensus sequences. They may have a core 6 to 8 nucleotide sequences. Almost 70% of them have similar sequences. Maybe only one nucleotide will change. That gives you less freedom for the software to look for exact string model. You say that base sequence is this. I am asking you, given sequence of several nucleotides, billions or millions of nucleotides, you give me exact match of it. of the sequences given. You go with exact string model, or you go with a string mismatch model. Okay, I might have an exact sequence, but you go with one mismatch, one variable mismatch. Give me all the sequences from the given sample. What are those sequences according to this? And then the software gives you an output for with one mismatch. And then degenerate string model. Again, we know that the promoter sequences, there may be upstream some 6 to 8 sequences close to them. There may be a consensus sequences of specific nucleotides, pyrimidines, or specific C's or G's. So you can define a specific given sequence here as S and D. S can be either C or G, D can be AGT coming out of these sequences. You can define them. The model can again based on the base sequence, based on the query you are putting, giving the freedom for the software to analyze. You can change that, then it gives an output. Another one called weight matrix model. The weight matrix model, given a nucleotide, given the stretch of 6 to 8, how many times, how many positions it is coming. It may be 0.1 and given a consensus sequences, all are T is 0.9 here. The A may be in other sequence 0.7, A is coming 3 out of 3. So you generate a score. That score also help you to define which base position the nucleotide promoter element contains. By doing so, you can get a peak, and you can define not only ATGCs, you can define different terms like pyrimidine, strong bonds, GC pair is important for primer design, and all but C, all but A, you can try all different combination of statisticals. N is anything, you can give the software. Another region of promoter integration is called microarray analysis. For example, the assumption is here is that co-expressed genes and regulated by common transcription factors, they may share a common regulatory elements in their promoters. Co-expressed genes, several genes in a given eye tissue, there are several genes that are involved for development of eye. They are expressed in a coordinated manner. So probably saying that same promoter elements are switching on different closely related genes in the function. So they may have a common transcription factor motifs in the digital elements or proximal elements. So basically, they may have a conserved promoters. That microarray analysis can be used for that. Some other algorithms are MEME algorithm, cis-regulatory module explorer. Further, they use same statistical models, Markov and hidden Markov model. And then another microarray analysis, Prima can be used for microarray analysis. Some of the promoter prediction, this fruit fly we have shown, ICG, another Tata box predictor, CSHL is Cold Spring Harbor Laboratory. They have a very nice promoter prediction software there. You can look into that. And a little bit more, if you are interested, further additional material, you can use for specific kind of software. You go into the published literature. You can search what the software, what it does, it give an outcome. You can look into that if you are really interested to work on bioinformatics in terms of genomics, and mainly in terms of cis-regulatory elements, leaving the transcription factors alone. You can go into specific software that can define only cis-regulatory elements. So in conclusion, you look into that promoter and ORF together. ORF can be used for gene prediction, but the promoter regions in terms of cis acting elements, you can define and predict a specific promoter of a given gene sequence or genome. Some of the slides I got it from Sarbesh T. Dangwal, one of the slide share presentation, very nicely done. You look into the effort the person has put, he has looked into several reference materials and looked and made the presentation into going into consolidated effort of these references. And then, okay, as we are coming to finishing of the class, let's thank all the frontline COVID workers and who are helping us. Hopefully the lockdown ceases and we will come back and resume the class.